So this week, basically we'll go through, I'll explain some of the differential media that we're using this week, and then after that, it's fairly straightforward. You'll be inoculating the plates and broths with your 4 organisms. The ELN this week is fairly straightforward as well, just basically describing and documenting each of these tests for each of your organisms and explaining how they work. And then after that, you'll have time to talk to your group, make sure that you have very clearly identified any resources that you still need, as we talked about in class this morning, so that you're ready to go next week. Right? And basically, the next couple weeks, almost the entire lab will be time for you to work on your projects. So we want to make sure that you're ready to do that and can make good use of that time. So this week, in terms of the media that we're using, a lot of them are based on the ability of a microorganism to degrade something, right? So we have hydrolysis of starch, casein, gelatin, DNA, lipids, and then we have a couple others that are kind of thrown in here. One is hemolysis of red blood cells. Again, it's a degradative, that's the word, right, process. And Kligler's iron agar, which is just a really cool test that looks at a number of different metabolic processes in microbes. A number of these are plates. A couple of them are in a different format. We'll talk about those as we get there. In some cases, breaking something down, you can actually just see a change in the media itself, as in gelatin hydrolysis and in casein hydrolysis. In other cases, we will add or have added an indicator that'll show the degradation products. Okay? So we'll just start here with starch. Starch hydrolysis. The media basically has some beef extract, which has amino acids and phosphorus and other things. And then the main carbohydrate source, however, is soluble starch. And basically, if organisms are capable of degrading the starch, which would indicate the presence of enzymes for that degradation, which are generally either referred to as glucosidases or amylases. The prefix amylo means starch. So if you took botany, you'll remember amyloplasts as another name for starch grains. Right? Amyloid plaques in the brain that we see if you have certain neurodegenerative diseases, they were thought originally to be starch. Turns out they're actually protein, but they stain similarly to starch when people were doing early histological studies on those neurological disorders, so the name got transferred over. But amylase, right, enzyme that breaks down amylo or starch, and it breaks it up into dextrins, which is just a name for short polymers of dextrose, which is another name for glucose, or glucose monomers. So it's either breaking it down into dimers or trimers or monomers of glucose. So how do we actually see that in the media? After you've grown an organism on this media for 24 to 48 hours, you actually are going to flood the plate with iodine. So you'll just take iodine and put it over the top, and you'll probably remember from Bio 125 or a high school course or other courses here at Dordt that iodine reacts with starch to give you this dark blue or dark brown color. And so what you'll actually see after you've done this, you'll see where you streak the organism, you'll actually see a clear halo around it, right, where there is no starch present anymore because the organism has these enzymes, these glucosidases or amylases, that have degraded it in proximity to where the organism was growing. So you'll streak the plates, you'll let them grow for 24 to 48 hours, come back, add the iodine, let it sit for a little while so the iodine can soak down into the media, and look for a halo. Right? There's a nice picture in your textbook of what this looks like, so you can see that if you're questioning your interpretation of the media. For this, you can use one plate for all 4 of your organisms, just as before, divided into quadrants, right? And identify which quadrant is which organism. Should be good to go. It's generally a fairly unambiguous result. The second differential media today is looking at the capacity of organisms to degrade casein. So casein is the primary protein in milk, and casein has a unique role. In addition to being a protein source, it's also what helps keep the lipids, or the milk fat, suspended in the milk, right? So if you make butter or something, you are going to try to disrupt the casein to release the fat, right? And then the fat will separate from the water, right, from the rest of the milk solution. That's what we're doing when you make butter. You can do it physically. You can also do it chemically by adding caseases, right? That will also release the fat. So what does this look like? If the plates look, maybe it's easiest just to see. Oh, we're still pouring those. We had a little trouble with them. Plates look white because they have milk in them, right? And maybe the question is, why does milk appear white? Right? The answer to that question is that lipids, right, and water have 2 different refractive indices for light. Right? So as light goes from the water into the lipid, back into the water, back into a lipid, back into a water, back into a lipid, back into the water, it's getting refracted. Right? It's getting split and bounced all over the place. And that makes the milk look opaque. Does that make sense? Okay. So if I start disrupting all of these little clusters, these little, what we call micelles, that have lipid inside of them and casein, if we disrupt those, the lipid will separate out it'll tend to separate out into its own little globules, and you'll have a clearer path for light through the milk, which will make it look more transparent. So again, you'll actually, if an organism is degrading the casein, if an organism is breaking open these little casein micelles, you're going to see a clear halo around where you streak the organism on the plate. Okay? Does that make sense? Right. So again, fairly unambiguous in terms of interpreting it. It's fairly easy to see whether an organism is positive or negative. Again, for this one plate, divided into 4 quadrants, that way you could do all 4 organisms on one plate. The third one today is gelatin hydrolysis. So gelatin is what you use to make Jell-O, right? It's a very small amount of gelatin. It's hydrated by a lot of water, which is why you can take a very little tiny bit of Jell-O powder and make a whole pan of, delicious jiggly stuff, right? Kids love to eat. But what is gelatin actually? Gelatin is actually partially denatured collagen, right? It's a protein that is a major constituent of our connective tissue. So we get Jell-O, we get collagen from things like the skin of animals, pig's hooves, other things like that. So maybe I just ruined that delicious jiggly dessert for you, but we certainly eat more bizarre things. I mean, what's one of the most delicious animal products we eat? Bacon. What is bacon? It's the belly fat of the pig, right? I mean, if that doesn't gross you out, I don't know why eating Jell-O would. But anyway, so in when you make this gelatin, when you make Jell-O, with gelatin, it forms this solid matrix. Certain organisms will have gelatinases, right, enzymes that can degrade the gelatin. If they do, it's going to break that gelatin down into smaller polypeptides, and you'll basically end up with a liquefied solution. And so these are poured as a tube, right? I don't want to take the cap off of that one. So you'll see it's fairly easy to interpret. Right now you have a solid, right? Solid media in there. You're going to stab inoculate this. And if the organism is capable of degrading the gelatin, basically if you tip this, it'll be liquid. Okay? So not hard to interpret. Right? 1 tube per organism, so you'll need 4. You'll need 4 of these tubes today. In there, besides the gelatin, are just a couple things to help support the growth of the organism: beef extract and some peptone. But fairly straightforward stuff. The 4th media we'll be working with today is DNase, DNase agar, and this has a couple of components to it. It has some tryptose, which is trypsin, using trypsin, which is a digestive enzyme in the stomach, to degrade a number of different things. But it's a general source of nutrition. DNA, so there's actually like just free DNA in the media, and then methyl green, which methyl green will associate with DNA when it's in a linear form, and then we have agar to create a solid substrate. And so the methyl green, when the DNA is intact, will create a green background. It's really quite pretty. Right? So this is our DNase agar. And again, basically what we're looking for, if the organism is capable of degrading DNA, the methyl green will not be able to associate with it anymore, and you'll get a halo, again, a clear halo around the organism that's capable of degrading DNA. So we're testing for DNases. We're testing for a particular enzyme, a cluster of enzymes capable of cleaving DNA. And in reality, most organisms have these, but most organisms don't necessarily secrete them into solution. Right? Internally, a lot of organisms have DNases that are responsible for recycling their own DNA. Right? But they don't necessarily have the equipment to degrade DNA in their environment. Fifth media that we're working with today is lipid hydrolysis media. The basic idea here, you have a plate with some general nutrition, beef extract and peptone, and then it has this unique compound in it called tributyrene. And tributyrene is a very simple triglyceride. Right? So we have our glycerol backbone, and then we have 3 butyric acid residues that are bonded through an ester bond, just like in other triglycerides. Like most triglycerides, this is a fairly hydrophobic molecule, right? It's like a really light oil. And the name here actually comes from this root, butyr, is butter. Right? So this stuff, when you degrade it, it smells bad because butyric acid, one of your byproducts, has a really kind of foul odor. The interesting thing is to really ask the question, what makes it foul? Other fatty acids are volatile, and we don't necessarily think that they smell bad. And it sort of begs the question, developmentally, why do we associate some smells as being good, right, or positive or enjoyable, and other ones as being, right, ugly or, But this is the smell that you'll get out of this from this volatile butyric acid is the smell of rancid butter. So it's not a good, I mean, most people don't enjoy it. So all we're really looking for here is the ability of microorganisms to cleave, right, these ester bonds between the butyric acid and the glycerol. How do we see that in the plates? When the media is poured and it has this tributylin in it, it tends to form these little fat droplets that you can see all in the media because this isn't oil, it's hydrophobic, it doesn't mix with the water in the plate. However, when you degrade it, right, when lipase acts on it, you end up with glycerol and butyric acid, both of which are water soluble. Right? So all of a sudden those little fat droplets are going to disappear. So you should be able to see in the vicinity of the inoculation, again, a clearing of these little fat droplets. Okay? So you can do one plate, again, for all 4 of your microorganisms. Just divide it into quadrants. Keep track of which one is which. The 6th media today is blood agar. Blood agar, in addition to being a differential media, is also used as a substrate for growing certain organisms that we call fastidious, that have very unique requirements, that are hard to culture in general. So there are some organisms, particularly a number of human pathogens, that only grow well on blood agar. Okay, they don't grow well just on general nutrient agar plates. Okay? So it's both a differential media and it's also a media sometimes used for fastidious organisms. As the name implies, it has blood in it. It also has some pancreatic digestive casein, right? So it has some protein in it and what we call beef heart infusion. So you blend up beef hearts and strain it, and that's basically what you got in the media as well. It's interesting to look at the history of how people came up with some of these media. Maybe we'll talk more about that in class sometime. What we're looking for here is the presence of certain hemolysins, and there's a whole bunch of different hemolysins, and there's a different capacity of microorganisms to carry out hemolysis as a process. Overall, though, that is, as the name implies, the degradation of red blood cells. And we tend to classify it into 3 types: beta, alpha, and gamma hemolysis. Beta hemolysis is complete hemolysis. The red blood cells are completely broken open. The heme is removed, right? The iron is being scavenged by these microorganisms. And you'll basically get a clear halo around organisms that are capable of doing beta hemolysis. Alpha hemolysis looks a little different. In this case, the heme can actually stay intact to some degree, but it may actually become reduced, and so you get this greenish sheen. So it's still very obvious, but it's not a complete clearing of the area around the organism where you inoculated it on the plate. Gamma hemolysis actually is non-hemolysis, so the agar still looks completely normal behind your organism. Okay. Any questions about any of these before we move on to Kligler's? Most of them are fairly straightforward. We're degrading a particular thing, and in some cases you can see that degradation directly. In a couple cases, we're adding an indicator to make it a little more obvious. Any questions? So streak, streak, stab, streak, streak, streak. Kligler's iron agar is a really super cool media. It actually can tell you a lot of different stuff about the organism that you inoculate it with. It's poured as a slant, and it has a combination of two media that we've actually worked with before. So the two we've worked with before, one is sim media, and the other is phenol red broths. So if you recall with sim media, we were looking at sulfur indole motility media, right? The ability of organisms to degrade cysteine, right? Produce hydrogen sulfide. The ability of organisms to degrade tryptophan, produce indole, and their motility. In this case, we're only, Kligler's has the S part, the sulfur part, okay? So it's capable of telling you whether or not an organism is capable of producing hydrogen sulfide. If you think back to that, does anybody remember what that looked like? What it was, what changed in the sim media that told you it was sulfur positive? Turned black. And so just like that, Kligler's iron agar, if it is sulfur positive, it will turn black, just like your sim media did. Okay? In addition to that, it has phenol red, which is a pH indicator, which you'll recall in the presence of a variety of fermentation end products, or particularly organic acids, pH in the media would drop, and that would cause phenol red to go from a red to a yellow color. Okay? So it's phenol red is a pH indicator, goes from yellow under acidic conditions up to a kind of a magenta color under alkaline conditions. And this is where it gets a little bit tricky. There's actually two sugars in here, glucose and lactose. But there's 10 times as much lactose as there is glucose. And so the way this works, glucose is generally very easy for microorganisms to use. It's usable by a much broader spectrum of organisms than lactose is. So initially, most organisms are going to use glucose, and they're going to grow, and you'll see either you'll be able to determine whether there's been aerobic growth or whether there's been anaerobic growth based on a pH change, just like we did with the phenol red broths. Okay? However, if an organism is capable of using glucose but not lactose, once this glucose gets used up, it's going to start using protein as a source of energy. And when it does, it's going to have to deaminate all those amino acids to use them as sugars. And the production of ammonia in the media is going to drive the pH up, right? It's going to make it more alkaline. So basically, if an organism is capable of metabolizing glucose but not lactose, initially you'll see it turn yellow, but then it'll turn purple, right? It'll go typically back to an alkaline state, drive the pH up towards, and that'll cause the phenol red to turn more of a magenta color. If, however, it can also use lactose, the organism will use the lactose instead of the protein, right? And that will maintain a yellow color. Does that kind of make sense? Okay. Now, there's something else really cool about this. Is it also tells us something about oxidation versus fermentation of those products as well. Because the way you inoculate this media is you're actually going to stab down into the base, and then when you pull the needle out, on the slant part, you're going to streak a fishtail up just like you would if it were a normal slant. And so the stab down into the bottom will create an anaerobic environment, in which case an organism would be fermenting the sugar, whereas in the slant portion, it would be carrying out aerobic metabolism of the sugar. So you can actually look at both the fermentation or the oxidation in the same tube, you can look at the fermentation and oxidation of both glucose and lactose in the same tube. And you're also able to identify, right, aerobic versus anaerobic growth, utilization or production of hydrogen sulfide. And the other fun thing that happens is because you did a stab, if the organism is producing gas as a result of the fermentation, which we saw before as well with the phenol broth, you had the little Durham tubes that would float or would capture gas. If you get gas production in the media, it will cause the media to split or pop off from the bottom of the tube. Right? So Kligler's shows you a whole bunch of stuff about your organism all in the same tube. Now there's a lot that I just said to try and interpret that. Your laboratory manual actually has a really nice table that will guide you through that interpretation. Okay. The important thing to remember, as I said, you have this glucose to lactose transition, which means it's kind of important to catch it at particular time points. It is time sensitive in a way that most of your other media hasn't been. So you may actually want to look at it several times over the course of the next 48 hours. Any questions about that? All right. Then those are our differential media. So you'll need 4 of those slants, 4 of the tubes, and then 1 plate of everything else. Get those inoculated. This is the last, as I mentioned before too, this is the last week of your knowns unknowns, so you don't have to carry them forward another week. You basically just need to inoculate these new media today and record your results. Again, with the ELN this week, please use a note card with your results, and make sure you add your results to the Google Docs spreadsheet, because after today, we should be able to match up knowns and unknowns. Any remaining time after you do that is for you to make sure that you are solid and prepared and have a plan to get started on your research next week. Sound good? Any last questions? All right, let's go to it.